Have you ever encountered claims that our very own DNA somehow, I don't know, disproves evolution?
Yeah, or that our genomes are like rapidly degrading, pushing us toward inevitable extinction.
Exactly. Today, we're taking a deep dive into the very core of these powerful and, let's face it, persistent arguments. Arguments that can sound quite compelling if you don't really look closely.
Right. And our source material for this deep dive is a fantastic essay by Skylar Fiction called DNA, Deep Time, and the Collapse of Creationist Genetics. It directly confronts common assertions from Christian apologists, specifically addressing claims made by Dr. Robert Carter of Creation Ministries International.
Okay. So our mission today is to meticulously unpack these claims against, well, the backdrop of modern genetic science.
Mm.
We're going to examine specific arguments about DNA's lifespan, mutation rates, how people interpret things like molecular clocks and DNA barcoding,
Mm-hmm.
and compare them directly with solid, peer-reviewed scientific findings.
Yeah, the goal here is really to help you separate the, let's say, scientific storytelling from the actual evidence-based understanding.
A shortcut to being well-informed on this topic, which can get pretty confusing.
Definitely.
So let's get right into it. Maybe starting with the core genetic claims that creationists often put forward.
Sounds good. So Dr. Robert Carter's central position, basically, is that genetics, when you properly understand it, doesn't support deep time or human evolution at all.
Right.
Instead, he argues the evidence confirms a much shorter biblical timeline, like a 4,500-year-old Earth, Noah's flood, incredibly rapid diversification of species after the flood,
Wow, Raph.
and ongoing genetic degradation ever since.
Okay, and he builds this on, I think the essay mentioned three main assertions. They sound quite definitive when you hear them.
That's right. First, the idea that DNA just can't persist longer than a few thousand years because it degrades so rapidly.
Okay, point one.
Second, that harmful mutations pile up so fast that long-term evolution is just impossible. That's the concept he calls genetic entropy.
Genetic entropy, right, got it.
And third, that things like DNA barcoding and molecular clock studies supposedly confirm all animal species, humans included, popped up within the last few thousand years.
Okay, three big claims. So which one do we tackle first? And maybe what's the most common misunderstanding people have about it?
Well, our source material really zeroes in on that second point first, genetic entropy. It's arguably the most popular creationist claim, but it's scientifically unsupported.
The idea that genomes are just sort of spiraling towards extinction.
Exactly. And the essay frames Carter's interpretation pretty directly as storytelling wrapped in scientific language. It suggests his arguments are based on, selective readings and untested assumptions, relying heavily on confirmation bias, really, rather than open scientific inquiry.
So the essay aims to provide a direct rebuttal using actual peer-reviewed science.
Precisely.
Okay, let's unpack that then. Is our genome really doomed by this genetic entropy idea?
Well, the claim itself, which comes largely from Dr. John Sanford and Carter adopted it, suggests that, yeah, human and all other genomes are rapidly deteriorating.
Because of?
Because of, supposedly, this overwhelming buildup of harmful mutations that natural selection is just too weak to remove, leading to, irreversible devolution. It sounds pretty dire, doesn't it?
It really does sound dire. But what's the scientific rebuttal? Is it just wrong?
It is, fundamentally. First off, the idea that most mutations are harmful is just a misrepresentation.
Oh.
Yeah, in reality, the vast majority of mutations are actually neutral. They don't really have a significant effect on an organism's survival or reproduction.
Okay, so most are neutral. But what about the ones that are genuinely harmful? How does life cope if errors keep happening? It still sounds like degradation could be a problem.
That's a great question, and that's where it gets really fascinating. There's this well-documented mechanism called purifying selection.
Purifying selection.
Think of it like genetic quality control. It effectively removes or weeds out those deleterious mutations over time. Even in small populations, it works.
Okay, so there's a cleanup crew.
Sort of, yeah. And then there are the beneficial mutations. Now, they're rare, definitely rare.
But they can significantly increase fitness, how well an organism does, and they can become fixed, meaning common, in a population, especially when there's some kind of selective pressure, like a changing environment.
So the profound insight here isn't just that genetic entropy is wrong, but that life, at its core genetic level is actually incredibly resilient. It's self-correcting almost.
Absolutely. It's a testament to this dynamic interplay of forces we often overlook.
It makes sense. Like, evolution wouldn't get very far if things just fell apart constantly.
Exactly. In fact, there was this really groundbreaking study computationally modeling this whole problem. And it showed that even rare beneficial mutations can effectively counteract the buildup of harmful ones. They can halt what's called Muller's ratchet.
Always ratchet. That's the idea that bad mutations just accumulate, like a ratchet clicking forward.
Precisely. Once they're in, they're hard to get rid of, supposedly. But this study showed beneficial mutations can stop that process. It means populations aren't doomed to just collapse genetically. They can adapt and persist, potentially indefinitely. And when you look at the math behind genetic entropy, it doesn't seem to add up either, right?
Not at all. Sanford's model, for instance, predicts human extinction thousands of years ago because of this supposed mutational load.
But clearly, we're still here. And not just here, but...
Right. Modern human populations show absolutely no evidence of collapse. Quite the opposite, actually. We see growth in numbers, increased longevity, incredible diversity.
So the prediction just fails the reality check.
Completely. And researchers have looked into this deeply. They've shown that even pretty high mutation rates can coexist with long-term genetic stability. Natural selection acts as this constant filter. As one paper put it, the amount of genetic variation you see is determined by this interplay of mutation, selection, and genetic drift, not by some inevitable degenerative buildup.
It's a dynamic balance, not a one-way street to doom.
Exactly, a dynamic balance.
So what does this all mean for us listening to these debates?
It means the appeal of genetic entropy seems to be more about its theological symmetry, you know? The idea that sin corrupted the world and mutations corrupted the genome.
Ah, I see the parallel.
But it's really a narrative, not validated science. It pretty much only exists within creationist literature. It lacks rigorous peer review or experimental testing in mainstream science.
It's a theory that hasn't really faced the music, scientifically speaking.
You could say that, yeah.
Okay, but if the genome isn't decaying like that, what about the sheer number of new mutations? That's another big claim from Carter, right?
Yes, that the human mutation rate, something like 100 new mutations per person, per generation, is just too high.
The argument being?
The argument being that humans couldn't possibly have survived for hundreds of thousands of years with that rate, so it implies a recent origin, maybe only 4,500 years ago, fitting that biblical timeline.
But again, this argument seems to overlook some key scientific details.
It does. Crucial nuance. As we mentioned, the vast majority of these mutations are neutral, or they happen in non-coding DNA regions. They don't significantly impact our fitness.
So they're just there, not causing problems.
Pretty much. And what's even more fascinating is that these very same mutation rates are why we can track ancestry across such incredibly long timescales. We use them for what we call molecular clocks.
Ah, so the mutations are actually useful for understanding history, not just a problem.
Exactly. They're like little ticks of the clock in our DNA.
Okay, so let's decode those clocks. What about the Y chromosome and mitochondrial DNA? Carter uses those too.
He does. He often points to what looks like a collapse in Y chromosome diversity as evidence for a bottleneck, like Noah's Ark reducing diversity.
Makes intuitive sense. Fewer men, less diversity.
It sounds plausible, but scientific studies clarify it's actually that purifying selection we talked about.
Ah, the cleanup crew again.
Right. It's particularly effective on the Y chromosome, removing harmful variants, and that's what primarily causes the observed low diversity, not necessarily a recent origin or a tiny bottleneck like the Ark.
Okay, so selection explains the Y chromosome pattern better. What about mitochondrial Eve? She gets brought up a lot.
Oh, constantly. And often misinterpreted by creationists as the only woman alive at some specific time in the recent past.
But the science says.
The science is very clear. She was simply the woman whose mitochondrial lineage, passed down only through daughters, happens to have survived in all humans living today. She wasn't alone.
She had contemporaries, other women whose lines just died out over time.
Exactly. And her scientific age, based on mutation rates in mitochondrial DNA, is estimated around 150,000 to 200,000 years ago.
Which fits squarely with the out-of-Africa model for human origins.
Perfectly. And it's far, far too old for biblical literalism.
And there's a similar figure for the male line, Y chromosome Adam.
Yep, the parallel concept. He's the most recent common male line ancestor. His estimated age is even a bit older, maybe 200,000 to 300,000 years ago.
And crucially, neither of them were the literal first humans.
Absolutely not. They are just common ancestors looking back through very specific genetic lines, maternal for Eve, paternal for Adam. It's like finding the single root of a very bushy family tree for just one type of inheritance.
Okay. And you mentioned earlier there's a methodological error in how creationists use mutation rates, extrapolating them backwards.
Yes, this is a key flaw in their argument. They often take the modern pedigree-based mutation rate, the rate you measure, by comparing parents and children directly, right now.
Okay, the short-term rate.
Exactly. And they try to project that rate backwards for hundreds of thousands, even millions of years.
But why is that wrong?
Because, as evolutionary geneticists explain, these pedigree-based rates tend to be higher than the long-term rates you calculate from comparing species over evolutionary time, the phylogenetic rates.
Higher.
It reflects differences in the timescales of selection. The short-term pedigree rate includes all mutations, even slightly harmful ones that haven't been weeded out yet by selection.
Selection hasn't had time to act on the very recent stuff.
Precisely. The long-term phylogenetic rate, however, reflects the mutations that have survived selection over millennia. It's naturally lower because the slightly bad stuff has been filtered out.
So using the short-term rate to estimate deep history inflates the number of mutations and makes the timeline look artificially short.
You got it. That's the methodological error. It's not a flaw in evolutionary theory. It's a misunderstanding of how mutation rates work over different timescales.
So the takeaway here is that human mutation rates actually demonstrate evolutionary resilience and a long history, not rapid decay or a recent creation event.
Couldn't have said it better myself.
This brings us to maybe the most direct challenge to the young Earth timeline, ancient DNA itself.
Right. The creationist position is pretty adamant. DNA just cannot survive more than a few thousand years. They use this to dismiss deep evolutionary timelines out of hand.
If the evidence can't exist, you don't have to deal with it.
Basically. But the scientific reality here, the breakthroughs in ancient DNA, well, they're truly remarkable. They completely upend that claim.
Okay, let's unpack this. What's the big example here?
The big one, highlighted in the essay, was back in 2013. A team led by Ludovic Orlando. They did something thought impossible.
Which was?
They sequenced a nearly complete genome from an ancient horse. And this horse wasn't just, medieval or something. It was preserved in Canadian permafrost.
Okay.
And dated to approximately 700,000 years ago.
Wow. 700,000 years. That's an order of magnitude older than previously thought possible for genomic data.
Exactly. They literally said it extended the time depth of paleogenomics by an order of magnitude. It was a massive breakthrough.
How did they even do that if DNA degrades? Wasn't it just dust?
Well, it wasn't intact chromosomes, that's for sure. The DNA was highly fragmented, chemically damaged, degraded.
So how?
They used incredibly sophisticated methods, targeting extremely short, overlapping sequences of DNA, sometimes just dozens of base pairs long, and then using powerful computers and modern horse genomes as references, they painstakingly pieced it all back together, like assembling a massive, shredded puzzle from tiny, faded confetti.
Incredible. So this highlights a really key point. DNA does degrade. Nobody denies that.
No, it follows known chemical processes.
But the rate of degradation depends heavily on the environment. Cold, dry, stable conditions like permafrost can preserve it for way, way longer than expected.
Absolutely. Temperature is the biggest factor. Freezing slows down the chemical reactions that break DNA down.
Yeah, the misunderstanding is thinking DNA has to be perfectly intact to be useful. Scientists are used to working with fragmented stuff.
That's right. They routinely work with degraded sequences. It's like archaeologists reconstructing a pot from broken shards. You don't need the whole pot intact to learn a lot.
Have they found even older or more degraded DNA since then?
Yes. The techniques keep improving. Studies have recovered identifiable DNA fragments, some as short as 25 base pairs, from ancient bones and even sediments in caves, sometimes pushing close to 1 million years or more under the right conditions.
So the idea that there's a hard 10,000-year limit or whatever number creationists pick is just wrong.
It's demonstrably false based on recovered data. The degradation is predictable based on chemistry, but it's not a universal rapid destruction within a few millennia.
And what's really fascinating here is that ancient DNA doesn't undermine evolution at all.
Quite the opposite.
It provides independent genetic confirmation. It lines up with the fossil record, helps refine our understanding of when species diverged, how they adapted.
It puts flesh on the bones of the fossil record, so to speak. It directly contradicts the few thousand years claim by providing tangible genetic evidence from the deep past.
It's like a genetic echo confirming that the deep past really happened.
A very powerful echo.
Okay, so ancient DNA is strong evidence. Let's tackle another area you mentioned, the misinterpretation of genetic convergence. particularly with mitochondrial Eve and DNA barcoding.
Right. Creationists sometimes use these concepts to argue for a recent origin, maybe within 6,000 years for all species.
And we already clarified mitochondrial Eve. Common maternal ancestor, not the only woman alive.
Exactly. And her age, 150,000 to 200,000 years ago, fits the standard evolutionary model, completely refuting a recent origin for humans.
So what about DNA barcoding? How does that get misused?
Well, DNA barcoding is primarily a tool for identifying species. You sequence a short, standard gene region, often in the mitochondria, and compare it. Species usually have distinct barcode sequences.
Like a genetic UPC code for species identification?
Kind of, yeah. Now, creationists often point to studies like one by Stokel and Thaler that got a lot of press, which found that the mitochondrial barcodes for many animal species seem to show relatively little diversity within the species.
Okay, low diversity within species. How do they spin that?
They argue that this low diversity and the clear gaps between species barcodes implies that all these species originated recently and simultaneously, maybe around the time of the flood.
Ah, linking low diversity directly to recent origin again.
Yes. But here's that crucial distinction we need to make, and it gets a bit subtle.
Okay, hit me.
The time it takes for the current mitochondrial lineages within a species to trace back to their most recent common ancestor, that's the coalescence time.
Right, the time they merge back to one ancestor, like mitochondrial Eve for humans.
Exactly. That coalescence time does not equal the time the species itself originated.
Okay, say that again. Coalescence time isn't species origin time. Why not?
Think about it. A species could be millions of years old, but maybe it went through a severe population bottleneck, say, during an ice age. Most mitochondrial lineages could have been wiped out. The survivors then repopulated, and all the current lineages would trace back to a common ancestor who lived relatively recently after that bottleneck.
Ah, so the coalescence time reflects recent demographic events, bottlenecks, rapid expansions, not necessarily when the species first appeared on Earth.
Exactly. Researchers like Ho and Log pointed this out clearly. Mitochondrial coalescence times reflect demographic history, not necessarily species age. Recent coalescence can reflect bottlenecks, not recent creation. It's a common pattern even in very old species.
So the creationist argument conflates these two different timescales.
Precisely. The essay calls it the creationist shell game. They swap lineage convergence time for species origin time, ignore the known effects of population history, all to make the data fit Genesis.
When in reality, these genetic markers, when properly understood, actually provide strong evidence for deep ancestry and complex population histories over vast timescales.
That's the scientific consensus, yes. It supports evolution, not recent creation.
Okay, so to really grasp why the creationist genetic arguments fail, we need to zoom out a bit, right? It's not just about DNA in isolation.
Absolutely. The essay emphasizes this strongly. It's about a whole constellation of independent dating methods that all consistently point towards deep time. DNA is just one star in that constellation.
And the big one, besides genetics, is radiometric dating.
The cornerstone of dating geological time, yes. It relies on the completely predictable decay of radioactive isotopes into stable isotopes.
Like a clock ticking inside the rocks themselves.
A very reliable clock. Each radioactive isotope has a known half-life, the time it takes for half of it to decay. These rates are constant, unaffected by heat, pressure, or chemical reactions.
And we use different isotopes for different timescales.
Right. Carbon-14 is famous for dating relatively recent organic materials, maybe up to 50,000 years or so, because its half-life is about 5,730 years. For older rocks, like volcanic layers, we use things like potassium-40 decaying to argon-40 with a half-life of 1.25 billion years. Or uranium-238 decaying to lin-206 with a half-life of about 4.5 billion years, which is how we date the Earth itself.
And the key thing is these methods aren't just operating in a vacuum, right? They get checked against other methods.
Oh, rigorously. They're validated and cross-checked constantly. For instance, carbon-14 dates are calibrated against tree rings dendrochronology going back thousands of years.
Tree rings, okay.
And longer timescales from radiometric dating are consistent with counts of annual layers in ice cores from Greenland and Antarctica, which go back hundreds of thousands of years. They're also consistent with layers in lake sediments, coral growth rings. All these independent systems give results that line up.
That consistency between unrelated methods is pretty powerful evidence for their reliability.
It's incredibly powerful. It's hard to argue they're all wrong in the exact same way.
And then there's the geological record itself, the layers of rock.
The stratigraphic record, yeah.
Yeah.
When you look at exposed rock layers, like in the Grand Canyon, you see this clear, consistent order of fossils.
Not just random jumbles.
Never random. You always find similar life forms in the lower, older layers. Then you get things like trilobites, then fishes, then amphibians, then reptiles, including dinosaurs in middle layers, and then mammals dominate the upper, younger layers.
Always in that predictable sequence, wherever you look on Earth.
Always. And this ordered progression is just completely incompatible with a young Earth or a single global flood. A flood would just mix everything up chaotically.
As geologists point out, these sequences show life forms evolving over millions of years, and the radiometric dates obtained from volcanic ash beds or igneous intrusions found within those layers confirm the ages.
The young Earth model just can't explain this ordered pattern without resorting to really strange ad hoc explanations, like animals sorting themselves by intelligence or mobility during the flood, which makes no geological sense.
So here's the really important question. What happens when these different clocks, the molecular clocks from DNA and the radiometric clocks from isotopes, talk to each other? Do they agree?
That's the crucial test. And the answer is yes. They yield strikingly similar timelines for major evolutionary events. They corroborate each other beautifully.
Like what? Give us some examples.
Okay. Emergence of anatomically modern humans. Both genetic data and the fossil record, dated radiometrically, point to around 200,000 years ago.
Okay.
The divergence of humans and Neanderthals, genetics and fossils consistently place it around 500,000 to 700,000 years ago. Go further back. Primate origins. Around 55 million years ago, based on both lines of evidence. The dinosaur extinction, bang on 66 million years ago, marked by an iridium layer dated radiometrically and confirmed by genetic divergence times of surviving groups like birds and mammals.
Wow. That convergence, that independent agreement across totally different fields using different methods, that's hard to ignore.
It's incredibly powerful evidence for the standard scientific timeline. Of course, anomalies sometimes pop up in dating.
Sure, science isn't perfect.
Right. Scientists understand these maybe inherited crystals in a sample or contamination. They have rigorous methods to detect and correct for these issues. They don't just throw out the entire methodology because of occasional outliers, unlike some critics.
So zooming back out, why does the creationist framework persist, even thrive in some communities, despite this mountain of converging evidence from genetics, geology, physics?
Well, the core issue, as the source essay really drives home, is that it's fundamentally not a scientific framework searching for answers. It's a theological commitment retrofitted with scientific language.
Meaning the conclusion comes first.
Exactly. It operates on circular reasoning. The conclusion, like a literal reading of Genesis, a young earth, Noah's flood, is fixed and non-negotiable. Then all data, all evidence, must be filtered, reinterpreted, sometimes distorted, or simply rejected if it doesn't fit that predetermined conclusion.
So if radiometric dating gives old ages,
Then radiometric dating must be flawed based on faulty assumptions.
if genetic data shows deep ancestry,
Then mutation rates must have been different, or we're miscounting, or maybe God created kinds with built-in diversity.
if ancient DNA is found from 700,000 years ago,
Then the sample must be contaminated, or the dating is wrong. The core belief is protected at all costs. It's a system designed to be immune to evidence that contradicts it.
which is, well, the exact opposite of how science is supposed to work.
Right. In stark contrast, evolutionary theory and the concept of deep time gains its strength precisely because of the convergence of evidence. You have genetics, geology, paleontology, archaeology, physics, chemistry, even linguistics, all pointing broadly in the same direction. Not because of some conspiracy, but because that's where the independent lines of evidence lead.
It's like multiple independent witnesses telling the same basic story. You start to think the story is probably true.
That's a good analogy.
And the essay points out there's a cost to rejecting this consensus.
There is. While faith and science don't have to be enemies, this specific approach of subordinating evidence to dogma, refusing to follow the data where it leads, it undermines public understanding of science. It can isolate followers from the broader world of scientific discovery and, frankly, teaches a distrust of objective inquiry itself.
So just to summarize the overwhelming scientific consensus based on all this evidence.
Okay. The Earth is about 4.5 billion years old. Life evolved gradually over billions of years. Humans evolved over millions of years, sharing common ancestors with other apes. Our own species, Homo sapiens, emerged around 200,000, 300,000 years ago in Africa. Our mitochondrial and Y chromosomal lineages trace back hundreds of thousands of years, not to single individuals, but to complex, interconnected population networks. DNA degrades predictably, yes, but under the right conditions, it can absolutely last for hundreds of thousands, potentially over a million years. We have the samples to prove it. And mutations aren't a fatal flaw, they are the fuel of evolution. They generate the variation that selection acts upon, allowing life to adapt, diversify, and persist. They refine life, they don't inherently destroy it.
No global flood needed to explain geology. No 6,000-year window for all of life's diversity. No impending genetic collapse. Just complexity, continuity, and immense time.
That's what the data points to, overwhelmingly.
So this deep dive into the source material, it really lays out a fundamental choice for you, the listener, doesn't it?
Yeah, it does. It's a choice between, as the essay puts it, clinging to an ancient story that struggles to accommodate new knowledge.
Or embracing a dynamic, unfolding story that's literally written in the code of life and the layers of the earth. The story the cosmos itself is telling us.
And the scientific understanding of deep time, evolution, genetics. It reveals a universe that's far older, stranger, and far more beautiful than maybe some narrow doctrines allow.
It means our existence isn't just a recent event, but it's tied to star stuff, literally, and billions of years of cosmic and biological unfolding. The source suggests that's not less meaning, but meaning magnified.
It reminds me of Carl Sagan's famous line, "The cosmos is within us. We are made of star stuff. We are a way for the universe to know itself." There's a profound beauty in that scientifically grounded view.
Absolutely. And the essay warns about the cultural cost of misinformation in this area.
Yeah, teaching distrust of data, mistaking rejection of science for intellectual strength. It can lead to what the essay calls intellectual bondage. It limits our ability to explore and appreciate the reality we actually live in.
So maybe a final provocative thought for you to mull over based on all this.
Think about this. What hidden truths, what deeper wonders about ourselves and our universe might we uncover? Not by trying to shrink the universe to fit our pre-existing beliefs.
But maybe by expanding our beliefs, our minds, to embrace the universe as it actually reveals itself to be, through evidence and inquiry.